Hello everyone, today we are going to discuss about introduction to embryology and gametogenesis. So these are the topic around which our discussion will turn around. So first moving on to the introduction to embryology. What is embryology? Embryology is the science that deals with the development and growth of an individual before birth. It starts with the fertilization and culminates with the birth of the baby. So the period of development from fertilization to the birth is called as prenatal development, and the prenatal period. This prenatal period is further divided into three periods, that is pre-embryonic period, embryonic period, and fetal period. Pre-embryonic period is a period that occurs from the fertilization to the second week of intrauterine life. What are the major events that occurs in the pre-embryonic period? The major event occurring in the pre-embryonic periods are formation of zygote, initiation of cleavage, implantation, and formation of bilaminar germ disc. Actually, bilaminar germ disc is formed in the it is an event of second week, and embryonic period exists from third week to the 8th week, and the major event occurring in the embryonic period is formation of placenta, umbilical cord, and extraembryonic membranes. So the differentiation of germ layer into the specific body organ. And in fetal period, it extends from 9th week to the birth. There occurs the growth and specialization of the body structure. Let's appreciate this from the figure. So we can see that from the fertilization up to the second week we have a pre-embryonic period. Embryonic period extends from third week up to the 8th week, that is embryonic period, and fetal period begins with at 9th week up to the birth of the baby. Talking about gametogenesis, what is gametogenesis? Gametogenesis is the process of formation of gamete, either male gamete or female gamete, from the primordial germ cell. So before learning about gametogenesis, we have a quick overview about the two types of cell division, mitosis and meiosis. What is mitosis? Mitosis is a type of cell division that occurs in the somatic cell. Here, one parent cell divide to give two daughter cell having equal number of chromosome as compared to the parental cell. So the stages that the different type of stages that occurs in the mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. What are the different events occurring in this phase? That is not topic of our concern, but let's have a quick recap over that. Prophase. In prophase, the chromosomes consist of two identical chromatids. We can see here chromosome containing two identical chromatids, one highlighted by blue and other highlighted by red, and chromatids are not recognized in this stage. We can also see the centrioles. Now these centrioles migrate to the opposite pole, move to the opposite poles, and chromatids become recognizable. We can see here they were closer apart, here they are far apart. Now these chromatids or chromosome line up on the equator. They line up on the equator, and a spindle fiber arises from the centrioles that gets attached to the centromere. So after that, the centromere is split and the chromatids becomes daughter chromosome. This individual chromatid becomes daughter chromosome and migrates to the either pole. Now, and nuclear membrane begins to reappear, and the cleavage for a start, and the cytokinesis occur. So after that, from one parental cell, it gives rise to the two daughter cell. So what is the significance of mitosis? The significance of mitosis include it maintains genetic stability, ensure continuous succession of identical cell. Similarly, regeneration, replacement, and repair of the cell is also due to the mitosis, and growth and development is also due to the mitosis. So, talking about the meiosis is a type of cell division that occurs only in the germ cell to produce male and female gamete. But initially, while forming the gamete, mitosis also plays a role. In the latter part, meiosis comes after. The meiosis consists of two stages, two cell division that occur subsequently one after another. They are meiosis 1 and meiosis 2. In meiosis 1, what happens that number of chromosome is reduced to the half of the mother cell or parent cell. Meiosis 2 is similar to the mitosis, but differs is that the DNA does not duplicate in this meiosis second. So let's talk about the different stages of meiosis. First, talking about these are the stages of meiosis 1, and these are the stages of meiosis second. And in meiosis 1, prophase is further divided into 5 sub stages, that is leptotin, zygotin, pacatin, diplotin, and dikinesis. What happens in the leptotin? In leptotin, chromosome appears as a cylinder threads. Each chromosome consists of 2 chromatids. In zygotin, there is a formation of bivalent, that is pairing of the homologous chromosome. And after that, in pacatin stage, 4 chromatids become visible. So 1, 2, 3, 4. That is called as tetrad. And in diplotin stage, there occurs a crossing over which leads to the formation of chiasma. Why crossing over takes place? Because of the exchange of genetic material. So in dikinesis stage, after the exchange of the genetic material, the chromosome migrate towards the nuclear membrane, that is opposite side. In metaphase, spindle formation occurs and the homologous chromosome gets arranged on the equatorial plane. And in anaphase, these chromosomes either two chromosome, one chromosome migrates to the other pole and other migrates to the opposite pole. That is, there is no splitting occurred as compared to the mitosis, in which splitting of the centromere were occurring, but here the chromosomes are divided into half, each migrating to the opposite pole. Now, in telophase, the cytokinesis occurs and the two daughter cells containing half number of chromosomes are formed. Here, after telophase, the second meiotic division occurs, and each daughter cell from the meiosis 1 give rise to two cell having the equal number of chromosomes. So we can see here one parental cell is giving rise to the 4 daughter cell having the half number of chromosome. Now, what is the significance of meiosis? The significance of meiosis is the maintenance of normal chromosomal number. Genetic variation is also due to the meiosis. We can see our crossing over is responsible for the genetic variation, and hybrid vigor is also due to the meiosis. Now, what is the difference between the mitosis and meiosis? The mitosis occurs in the somatic cell, whereas meiosis occurs in the germ cell. Mitosis completes in one sequence, whereas meiosis completes in 2 sequences. There is no crossing over of chromatid in the mitosis, whereas crossing over of chromatids occur in meiosis. Same number of chromosome in daughter cell as parent cell in mitosis, whereas there is a half number of chromosome in meiosis. Daughter cells are identical to each other and to the parental cell in mitosis. We can say clone. And in meiosis, daughter cells are not identical to each other and to the parental cell, whereas mitosis is called as equational cell division as the number of chromosome is not reduced, but meiosis is called as reductional cell division because the number of chromosome is reduced here. Now, talking about the spermatogenesis. What is spermatogenesis? Spermatogenesis is the process of formation of spermatozoa from spermatogonia. Now, how this spermatogonia is formed? It is formed from the primordial germ cell, and that process is called spermatocytogenesis. The process of formation of spermatogonia from primordial germ cell is called as spermatocytogenesis. What is the time When spermatogenesis occurs, it begins only during the reproductive period, unlike that of female, and the process of formation of oocyte occurs before birth, but in male it begins only during the reproductive period at the age of puberty and continues even into the older age. What are the total duration that takes place to form the spermatogonia? It is about 64 to 74 days. So let's have an overview about spermatogenesis. In spermatogenesis, primordial germ cell undergo spermatocytogenesis to form spermatogonium. This spermatogonium is light and dark, type A, type B, which we will discussing in the subsequent slide. But that spermatogonium, especially type B spermatogonium, undergo mitosis from primary spermatocyte consisting of 44XY chromosome. Primary spermatocyte undergo first meiotic division to form secondary spermatocyte, and secondary spermatocyte further undergoes second meiotic division to form spermatid, and spermatid undergo spermiogenesis. So rounded cell is converted into the tadpole-like cell, that is spermatozoon. So let's discuss about in detail about the spermatogenesis. In spermatogenesis, primordial germ cell undergo mitosis cell division to form dark type A spermatogonium 44XY 44XY. This dark type A acts as a stem cell which acts as a stem cell. That dark type A spermatogonium is converted into light type A spermatogonium and another dark type A spermatogonium. Then light type A spermatogonium is converted into type B spermatogonium and type B spermatogonium to type B spermatogonium. Now this type B spermatogonium undergo mitotic cell division to form primary spermatocyte, which is the largest germ cell. This primary spermatocyte undergoes first meiotic division to containing half number of chromosomes than the primary spermatocyte and is called as secondary spermatocyte. This secondary spermatocyte further undergo meiotic cell division, that is equational cell division to form spermatids, which is the rounded cell, and they further undergo spermiogenesis to form spermatozoa. Now in this is the picture taken from the Langman's embryology. We can see here up to the early late spermatids, the cytokinesis we have learned that in mitosis cell division karyokinesis is followed by cytokinesis, but in spermatogenesis the cytokinesis remains incomplete, due to which the cell remains attached to each other up to the stages of late spermatid, and after the removal of the residual body spermiogenesis, the cells get separated or spermatozoa gets separated. Now talking about the structure of a spermatozoon, why we are more concerned about the structure of a spermatozoon? Because we are next few slides we are talking about the spermiogenesis. We are going to discuss about the spermiogenesis. So to learn about spermiogenesis, we first need to be oriented about which organelles or which structure is present at which parts of the mature spermatozoa. So the spermatozoon, the length of the spermatozoon is 50 micron, and the parts include head, neck, and tail. The tail is further divided into middle piece, principal piece, and end piece. Head, talking about head is sphere head in cross section. In section, it consists of nucleus, and nucleus is anterior 2/3 of the nucleus is covered by the exosomal cap. That exosomal cap is developed from the Golgi apparatus. We will talking about in the subsequent slide. This exosomal cap consists of hyaluronidase and acrosine enzyme. What is the function of hyaluronidase and acrosine? You can comment down below, but we will discuss in the next video. Neck is narrow. It consists of basal plate which is funnel shaped, and centriole, which gives rise to the axial filament. Tail consists of middle piece, principal piece, and end piece. That is middle piece, principal piece, and end piece. What are the difference in the structure of middle piece, principal piece, and end piece? In middle piece, there is a centrally located axial filament which consists of cilia in a 9+2 arrangement, which is surrounded by the 9 outer dense fiber, and which in turn is surrounded by the mitochondrial sheath and then cell membrane. But in principal piece, here we can see it is rounded in cross section. It is more or like oval in cross section. It consists of the axial filament in the center, which is surrounded by the 7 outer dense fiber, and which in turn is surrounded by the fibrous sheath and the cell membrane. In end piece, there is only axial filaments are present. There is no outer dense fiber and cell membrane are present. What is the function of axial filament? The function of axial filament is the movement of spermatozoa. So, talking about spermiogenesis, what is spermiogenesis? Spermiogenesis is the process by which spermatids are transformed into the spermatozoa. Process is known as spermiogenesis. What are the major changes that occurs in spermiogenesis? That is nuclear morphogenesis and condensation. Nuclear morphogenesis and condensation, which gives rise to head and formation. There will be formation of tail. Why tail is formed? Because to provide movement. Similarly, acrosome is formed, which contains enzymes like hyaluronidase, acrosine. It is a rearrangement of organelles like mitochondria, centrioles also occur, and excess of cytoplasm is thrown out, or shedding of the excess cytoplasm takes place in the spermiogenesis. So what are the different events? Let's appreciate it from the figure. Here in spermatids, here we can see Golgi apparatus surrounded and the central nucleus containing centrosome at the periphery, and mitochondria is also present. Now this nuclear material chromatin gets condensed and moved towards one pole, which give rise to the head. Now the Golgi apparatus, which is shown here in yellow color, forms the acrosomal cap that covers the anterior 2/3 of the nucleus. Similarly, centrosome divides into two. And one centrosome lies at the area of neck and forms axial filament, while other forms the annulus at the distal end of the middle piece. Similarly, the organelles like mitochondria gets also arranged around the middle piece of the tail, and excess of cytoplasm is thrown out from the spermatids, and in this way finally we get a mature spermatozoa. This is the process of spermiogenesis. Now let's appreciate this process again in a more clear way. What happens in a spermatid, which was initially rounded cell, nucleus forms the head. Similarly, Golgi apparatus forms the acrosomal cap, and one centrosome divides into two centriole. One lies in the neck and forms the axial filament, and other forms the annulus at the distal end of the middle piece. We have already discussed. And mitochondria surrounds the axial filament spirally between the neck and middle piece to form the between the neck and annulus to form the middle piece. Similarly, cell membrane is intact. Now this is the picture taken from clinical embryology by Inderbir Singh. This is also saying that telling the same story, that is the Golgi apparatus forms the acrosomal cap, nucleus forms the head, proximal centriole lies in the neck, distal centriole becomes ring shaped and forms the annulus, mitochondria form the sheath of the middle piece, and axial filament grows outside the centriole, present in the middle piece and tail piece. So this was all about the spermiogenesis. Now talking about the clinical correlation, abnormal sperms. There can be morphological abnormality, or morphological abnormality includes a sperm where head and tail may be abnormal. There can be two head as shown in this figure, or two tail that shown in the figure. Similarly, a sperm may be too giant or dwarf, as we can appreciate by this. And Similarly, the sperms may be dwarf. Similarly, the sperm may contain double body, and it is a normal spermatozoa. Now, another abnormal sperm has immotility. For potential fertility, 50% of sperm should be motile even after the 2 hours of ejaculation. If less than 50% are motile, then they may lead to the infertility. Similarly, there can be genetic abnormalities like non-disjunction and other, which we will discuss at the end of this chapter. Now, talking about oogenesis, by a sequence of events by which oogonia are transformed into the secondary or mature oocyte is known as oogenesis. Here, primordial germ cell within the ovary divides by mitosis to form a large number of oogonia. I haven't explained how primordial germ cell have appeared in the gonads of the either male or female. We will discuss it in detail in the coming chapters of development of reproductive system. So what happens in oogenesis? The primordial germ cell, which consists of 44 XX chromosome, is converted into oogonium, and this oogonium enlarges to form primary oocyte. That primary oocyte undergo first meiotic division to give secondary oocyte and first polar body. Here the division is unequal cell division. Similarly, secondary oocyte undergo second meiotic division to form second ovum or secondary oocyte and other polar body. Similarly, the first polar body also undergo second meiotic division to form two polar body. Primordial germ cell within the ovary divides by mitosis to form a large number of oogonium. Each oogonium then enlarges to form a primary oocyte. The primary oocyte enters the prophase of the first meiotic division, which is enters the prophase of the first meiotic division before birth, but that meiotic division is prolonged up to the puberty. Why? Due to the oocyte maturation inhibitor, which is secreted inhibitor factor secreted by the follicular cell surrounding the oocyte. The first meiotic division completes just before the ovulation. This meiotic division is unequal. Here, majority of cytoplasm goes to the one daughter cell, which forms the secondary oocyte, whereas the other daughter cell barely receives any cytoplasm and gives rise to first polar body. We can also see here the division is. Similarly, the secondary oocyte enters the second meiotic division at the time of ovulation, and it completes the second meiotic division at fertilization. The second meiotic division is also unequal, giving rise to mature oocyte and other second polar body. Here, the only one daughter cell receives most of the cytoplasm, which gives the mature oocyte. The first polar body also undergoes second meiotic division and gives rise to two additional polar body. And this was all about oogenesis. We will be discussing it in detail in the upcoming chapters of female reproductive cycle, about different types of oocyte at different stages of development of female fetus. But let's have an overview of a timeline and event taking place during the oogenesis. At 3-month prenatal period, the cell type present in the ovary is primordial germ cell, and the event that takes place is that primordial germ cell migrate from the yolk sac to the gonadal ridge. So don't get afraid by the term of gonadal ridge or yolk sac. We will be discussing it in the coming days. Similarly, 5-month prenatal period, the number is about 7 lakh, and the primary oocyte are enclosed by surface epithelial cells of the gonadal ridge. And what are the event takes place? That lots of mitosis and some oocyte started meiosis 1 and arrested in prophase. Similarly, 7-month prenatal period, where cell type is primary oocyte, is surrounded by the follicular cell, which form the primordial follicle. Here, the degeneration with loss of germ cell oocyte is also present in the prophase. Only at birth, the number is about 6 lakh to 8 lakh approximately, and the cell type is primary oocyte and primordial follicle. All oocyte enters the meiosis first and arrest in the diplotin or prophase. First oocyte lies towards the surface of the ovary in this process. Similarly, in puberty, 40,000 out of which only 500 ovulate during the reproductive span. And what are the majority cell type during the secondary oocyte? Complete meiosis 1 just before the ovulation, and the release of the polar body takes place. Meiosis 2 is incomplete and stops 3 hours before ovulation. Similarly, fertilization, the number of 1 second oocyte gets chance to ovulate, and the cell type present here after fertilization is zygote, and the fertilization of secondary oocyte occurs at the time of secondary oocyte occurs with a sperm, and the second meiotic division is completed. These are the different cells, that is primordial germ cell, similarly oogonium, and primary oocyte in the prophase, arrested in the prophase. Similarly, at the 4 month we can see the flat epithelial cells and oogonia surrounded by the flat epithelial cell. Some of the primary oocyte are in the prophase of the first meiotic division. At around 7 month, the primary oocyte is surrounded by the follicular cell, and they are present in the prophase. Similarly, in newborn, the resting primary oocyte is present at the diplotin stage and surrounded by the follicular cell. Now, talking about the various stages of development of ovarian follicle, the various stages are primordial follicle, primary follicle, secondary follicle, and Graafian follicle. Primordial follicle, the oogonium gets covered by a single layer of flat epithelial cell, which is called as follicular cell, which are derived from the stromal cells of the ovary or surface epithelium of the ovary. This is called as primordial follicle. It consists of a single large nucleus with prominent eccentric nucleolus. Similarly, primary follicle, the flattened layer of follicular cell becomes columnar and forms a unilaminar primary follicle, and the follicular cell proliferate to form several layers for the formation of membrana granulosa cell in the maturing primary follicle. These membrana granulosa cells secrete primary oocyte and its granulosa cells secrete a glycoprotein-like substances that forms a thick homogeneous membrane between the granulosa cell and primary oocyte, which is called as zona pellucida. Similarly, the granulosa cell rests on the basement membrane that separates these cells from the surrounding stromal cell. This is called as maturing primary follicle. Now, talking about the secondary follicle, a small fluid-filled cavity appears between the follicular cell. This cavity fuses to form an antrum, so it is also called as secondary follicles, maturing secondary follicle, or antral follicle, or vesicular follicle. Now, the antrum gradually increases in size. It increases in size and it pushes the oocyte towards one side of the follicle. The granulosa cell that surrounds the oocyte towards one side is known as cumulus oophorus, and those that attach the oocyte to the wall of the follicles are called as discus proligerus. So, talking about as the follicle expands, the stromal cell surrounding the granulosa cell become condensed to form a covering, which is called the theca interna. Outside the theca interna, some fibrous tissue some fibrous tissue gets condensed to form another covering, which is called the theca externa. So this hole, that is the secondary oocyte surrounded by the follicular cells called cumulus oophorus, oophorus containing antrum, and the stromal cell forming theca interna and theca externa. This hole is called as a Graafian follicle. The Graafian follicle, this hole is called as Graafian follicle. All here what we have to note that all ovarian follicle, be it primordial follicle, primary follicle, secondary follicle or Graafian follicle, it consists of primary oocyte. The primary oocyte completes the first meiotic division and from secondary oocyte and first polar body. The difference between the oogenesis and spermatogenesis. The oogenesis, primordial germ cell, talking event wise, primordial germ cell and gonad time migrates from the yolk sac to the gonadal ridge in the second to third month of intrauterine life. So oogenesis occurs only ovary in the absence of testis determining factor. Similarly, spermatogenesis takes place in testis. Talking about the meiosis at the time of birth, the primary oocyte is arrested in the diplotin stage of the prophase of meiosis first under the influence of ovary oocyte maturation inhibitor factor, whereas spermatogenesis at the time of birth, primordial germ cell and Sertoli cells lines the seminiferous tubules. No spermatids or anything is formed at the time of birth. At puberty, ovulation begins and primary oocyte divides to form secondary oocyte. Similarly, primary spermatocyte forms secondary spermatocyte at the time of puberty. It forms spermatids that undergo spermiogenesis to form mature spermatozoa. Similarly, final completion of meiosis second will happen only at the time of fertilization. Here in spermatogenesis, entire process of meiosis is completed without any arrest. Where that is the difference. Here the division is arrested at a particular time before birth and before fertilization, but it is a continuous process in spermatogenesis. Similarly, the location of oogenesis, it occurs inside the ovary, but the completion of meiosis second happens in the fallopian tube. Similarly, spermatogenesis occurs inside the testis. Similarly, talking about the cell division is unequal in oogenesis, whereas it is equal in spermatogenesis. Now, talking about the end result, what are the end result of oogenesis? The oogenesis give rise to one haploid ovum and 3 polar body, whereas in spermatogenesis we get 4 haploid sperm, polar body. Similarly, oogenesis produces non-motile cell, whereas spermatogenesis produces motile cell. Actually, structure is seen in the oogenesis, whereas cell restructuring is seen. So you can guess here what is it pointing towards. Cell restructuring, that is, it is pointing towards spermiogenesis. Reduction of cytoplasm and nuclear condensation occurs in the spermatogenesis. Similarly, the size of ovum is larger than that of the oogonium in oogenesis, whereas the size of spermatozoa is smaller than that of spermatogonium. The lifespan or time span taking for the oogenesis is, it begins his fetal life and then arrested until puberty, resumes at puberty and continues until menopause, but begins at the puberty and continues throughout the life in spermatogenesis, that is, it started late and continued throughout the life. Similarly, it is a cyclical process once a month, that is talking about the ovulation. Similarly, spermatogenesis continues on daily basis. Approximately 500 oocyte ovulate during the reproductive time span, but here millions of spermatozoa develops 2 to 3 million release in each ejaculation. This was all about the oogenesis and spermatogenesis. Now talking about the applied aspect, we have come about the genetic abnormalities, which is mainly related to the cell division. So the chromosomal abnormalities can be numerical and structural. So talking about numerical, first of all, let's talk about what are the normal somatic cell. Normal somatic cell consists about the 46 chromosome, that is diploid, whereas normal gamete contains 23 chromosome, that is haploid. So what is euploid? Euploid means exact multiple of n, that is 2n, 3n. For example, let's assume the value of n as 20. 23. So 2n maybe will be 46, that is diploid. 3n will be triploid. Similarly, what is aneuploid? Aneuploid means that is word is itself explaining that it is not euploid. So here either extra chromosome is present, that is trisomy, we have this talked in Down syndrome, trisomy 21, or one chromosome is missing, which is called as monosomy. Similarly, abnormalities during the meiotic or mitotic cell division, that is non-disjunction, is also takes place. Similarly, translocation such as balanced and unbalanced translocation gives also rise to chromosomal abnormality. What is balanced translocation? If the translocation occurs but that does not cause any produce any clinically significant symptoms, then that is balanced translocation. But if it's produce a clinically important significant symptom, then that is unbalanced translocation. We will be discussing in the subsequent slide. Similarly, structural change, that is may be chromosomal breakage. One of the brilliant example is cri-du-chat syndrome. What happens in cri-du-chat syndrome? There occurs the partial deletion of the short arm of the chromosome number 5. Similarly, microdeletion on the long arm of chromosome 15 give rise to two syndrome, that is Angelman syndrome and Parder-Willi syndrome. Then what is the foundational difference in between these two syndrome? The difference is that Angelman syndrome occurs in the microdeletion on the long arm of 15 chromosome of maternal chromosome. Similarly, Parder-Willi syndrome occurs due to microdeletion on long arm of 15th chromosome, which is derived from the paternal side. Now let's discuss about the cri-du-chat syndrome. Cri-du-chat syndrome is also called as cat's cry syndrome. This condition is caused by a deletion in the short arm of chromosome 5. What are the characteristics that occurs in the cri-du-chat syndrome? The affected individual have round face and they have characteristic cats like cry. Mewing cry of a child is also seen. Similarly, so let's discuss in brief about the different types of syndrome. What happens in Down syndrome? The incidence of Down syndrome is 1 in 700 birth. The trisomy 21 type, the karyotype may be trisomy 21 type, that is consisting of 47 chromosome XX plus extra 21 chromosome. There may be translocation type, that is 46XX that is translocation between 14-21. Similarly, mosaic type may also be present, 46XX or 47XX plus 21. What are the characteristics that occurs in the Down syndrome? We can talk about there is a intellectual disability. Similarly, abundant neck skin is seen. Similarly, epicanthic fold and flat facial profile is also seen. Similarly, in hand we can see simian crease and congenital heart defects like heart defects are also present. Similarly, intestinal stenosis, umbilical hernia, they are predisposed, high risk of predisposition to leukemia. Similarly, hypotonia, loss of muscle tone, lack of muscle tone, and there is a gap between the first and second toe is also present in the Down syndrome. Now talking about the Edwards syndrome, the incidence is 1 in 8,000 live birth. The karyotypes may be trisomy of 18, 47XX, that is 18th chromosome is extra here. Similarly, mosaic type, that is 46XX or 47XX with extra 18 number of chromosome. So what are the clinical features of the child having Edwards syndrome? They have prominent occiput. Similarly, intellectual disability is present, microgenetia is present. Similarly, low set ears are present. Similarly, they have short neck, overlapping fingers are present. Similarly, they are also at the risk of having congenital heart defect, renal malformation. Here what here in it is saying renal malformation is also seen. Here in figure we can appreciate the horseshoe shaped kidney. Similarly, there is a limited hip abduction movement, and rocker bottom feet is also present in the Edwards syndrome. Now talking about the Patau syndrome, it is trisomy of 13 and the incidence is 1 in 15,000 baht. So karyotype is trisomy 13 type is 47XX. It consists of extra 13 chromosome, and the translocation type is 46XX with extra 13 chromosome, and mosaic type is 46XX or 47XX plus 13. Similarly, the clinical features of the affected child consist of microcephaly and intellectual disability. Similarly, they have microophthalmia, polydactyly, extra finger, cleft lip and palate are the characteristic feature. And similarly, renal defect is also seen. Cardiac defect, what here we can see polycystic kidney, we can appreciate here. Similarly, rocker bottom feet is also seen in Patau syndrome. So in Turner syndrome, it is a monosomic condition where extra copy of X chromosome due to the loss of one X chromosome takes place due to the non-disjunction during the meiosis. Similarly, the incidence is 1 in 3,000 female birth. And what are the different clinical features of the child suffering from the Turner syndrome is short stature. They have congenital heart defects like coarctation of aorta. They have low posterior hairline. Waving of neck is also present. Similarly, they have broad chest and widely spaced nipple, and cubitus valgus is also present, which is bilateral. And now talking about what is cubitus valgus. Similarly, they have gonadal dysgenesis and are associated with the amenorrhea. Similarly, pigmented nevi is also present. And talking about they have peripheral lymphedema at the birth. So we will be discussing this genetic defect in detail in the upcoming chapters of the medical genetics. Klinefilter syndrome, it is a trisomic condition. It is found only in male. It is caused by dysfunction of the XX chromosome during the gametogenesis. As a result, chromosomal complement in the somatic cell is XXY. So what happens in female? If the non-disjunction takes place, the resulting gamete consists of one less number of X chromosome, and that gives rise to Turner syndrome. But if the non-disjunction takes place and in the male gamete, and that male gamete consists one extra copy of X chromosome, then that will give rise to the Klinefilter syndrome. So the individual is phenotypically a male, but consists of sex chromatin, that is bar body positive due to the extra copy of X chromosome. The characteristics of the affected male are similarly they have tall stature, slightly feminized physique. They have mildly impaired IQ, consists of 15 point or less than average, absent or delayed or incomplete puberty, less muscle compared to age, matched control. Similarly, osteoporosis is also, but they have characteristics like learning disability. You can read it from here. And now and the incidence of Klinefelter syndrome is 1:500. Now talking about the two important syndrome like Angelman syndrome and Parder-Willi syndrome, what happens? It is a 15th chromosome. If the microdeletion occurs in the maternal chromosome, then that gives rise to the Angelman syndrome. Whereas if the microdeletion occurs in the paternal chromosome, then it gives rise to the Parder-Willi syndrome. The features of the affected child with Parder-Willi syndrome is hyperphagia, insatiable appetite, and obesity. Similarly, they have short stature with a small hand and feet, hypotonia, hypogonadism, mild to moderate mental retardation, and behavioral problem such as rage and violence is also present. Now talking about the Angelman syndrome, they have a happy clinical feature is happy disposition with inappropriate laughter. They have severe mental retardation, that is IQ is less than is up to 5 to 10. Now they have ataxic gait. Ataxic gait may be stiff, jerky, unsteady, and seizure are also present. So this is the pictures of a child who is suffering from the Angelman syndrome. Now that's it from my side. Let's have a quick view about the some clinical problem. You can pause the video and look over it. The answer of this clinical problem is have been discussed or have been written in the notes which is provided in the description of this video. So check it out over there. Thank